So with a single molecule of glucose, how many NADHs total could I produce using the citric acid cycle?
6.
6. How many molecules of ATP could I produce? 2. How many molecules of FADH?
2.
2. Okay. So notice at this point in time, we've only made 4 molecules of ATP net between glycolysis and the citric acid cycle. But how many molecules of NADH have we made? Right, 2, 4, and then if there's another 6 here, that gives us 10. 10 molecules of NADH and 10 protons, along with FADH2. Questions, concerns, points of clarification?
I thought
1 for every revolution of the cycle, which means a molecule of glucose produces 2 pyruvate, which can be converted into 2 acetyl-CoAs, which give us 2 revolutions of the cycle, which gives us 2 FADH2s. Richard?
I thought that glucolysis and the citric acid cycle produce the same amount of ATP.
They do?
They do. Okay.
Yeah, they do
Oh.
make the same amount.
Okay, so we're not losing ATP on the citric acid cycle like what we do on the glucolysis.
Right. Correct.
All right.
Yes, Stephanie?
Just double checking, every time NADH is produced, electron produces
Yes, that is correct. Very, very important part of that particular reaction. Okay. So, I guess I'll have to finish fermentation when we meet again with the start of photosynthesis. But we're going to get through oxidative phosphorylation because there isn't much to it. But the stuff there is always seems to be very, very dense. So, why, you may be asking yourself, have we been making so much of these electron donors, this NADH and that FADH2? And why do I keep going off like a crazy person about how they're so important and why we need to have all these protons? Now I'm finally going to show you how this is all going to pay off and why I'm so utterly excited about oxidative phosphorylation. Okay. What we're going to do is we're going to use the electrons that these molecules are carrying to make an electrochemical gradient that pulls protons towards it and through a membrane. And we're going to use these protons to make the ATP. Okay? That's where the electron transport chain comes in, and that's also where a very, very, very important enzyme comes in. Okay? So we're going to use the electron transport chain. This is where we're transferring electrons from molecule to molecule. And as we transfer those electrons, we slowly decrease the total amount of potential energy that they have. Okay? That energy that's being released is what we're going to use to pump those protons to one side of the membrane and create a concentration gradient. Just so you know, at the very end of the electron transport chain, the final place that those electrons go is oxygen. Oxygen accepts the final 2 electrons, and in doing so, it attracts 2 protons to form water. Hence the reason why it's called oxidative phosphorylation. Oxygen is the final electron acceptor. It's also why it's called aerobic respiration, because oxygen is present. And I'll show you oxygen becoming water in the diagram. Okay? So here's the electron transport chain. Basically, it's a series of proteins in the inner membrane of the mitochondria that transfer electrons from protein to protein to protein to protein. As they do that, they generate this chemical gradient, where these are going to be very negatively charged, attracting the protons and pumping the protons through some of these pumped proteins or structures. At the very end of the chain, the final 2 electrons are going to be transferred to a molecule of oxygen, which then combines with 2 protons to form a molecule of water. This is pretty much just a basic idea of what's going on, start to finish. Questions or concerns? I'll understand if you have.
Repeat the last part about the final 2
Yes, the final two electrons of the electron transport chain are accepted by oxygen, and it combines with protons to form water. Is that what you were going to ask too, Katie? Okay. What's up? Okay.
You said it's going to be negatively charged all the way through.
Well, if you think about it, we're moving electrons from one molecule to the next. What's going to happen to a molecule that accepts electrons?
Becomes negatively
It's gonna be
charged.
negatively charged, right? That's what we're going to use to pump those protons through. We're just going to have a bunch of electrons coming in here with all these negative charges that they're going to start attracting those protons. Remember, Paul Abdul, opposites attract. Coming right back to where we started. Okay. So there are this abundant number of proteins, including things like cytochromes, that are going to accept these electrons and transfer them through. The reason why it's so important that they do this is they're going to then pump the protons through, generating a gradient. Please note that the electron transport chain itself does not produce ATP at all, period. No ATP is produced when we're pumping those protons through the electron transport chain's proteins. None at all. This is very confusing because in your book they show you a picture where it has those little like water wheels that represent the electron transport chain, and it's got a collective arrow that comes out and it says ATP. I don't know why they wrote the book that way, because it then makes you as a student think, oh, the electron transport chain is what makes ATP. It's not. What actually happens is we pump all of those protons through. So here is my membrane. Here are my proteins involved in the electron transport chain. And on the other side here, I'm going to have this specialized protein, which is called ATP synthase. It's an enzyme that synthesizes ATP. Here's what's going to happen. My NADHs and my FADH2s are going to transfer their electrons, becoming FAD and NAD+ when they do it. Those electrons are going to move through the chain, eventually being donated to oxygen so that it can form water. With those protons. But while they're doing that, protons are attracted. If I draw them properly. Protons are attracted to the negative charges that all of these proteins are now exhibiting because we're pumping electrons through them, because the electrons are being transferred. So the protons are going to be attracted to these proteins. These proteins act as a channel where those protons are literally pumped through the proteins, giving us a ridiculously high proton concentration on the opposite side of the membrane. So we've got all these protons on this side of the membrane. If I were to ask you if those protons could permeate the membrane, which direction would they diffuse?
Out.
In. Why? Higher concentration to lower concentration. So here is why I love oxidative phosphorylation. ATP synthase is a protein that allows protons to diffuse back across the membrane, but as they diffuse through it, it uses their natural diffusion down their concentration gradient as a means of making ATP. So this protein turns as these protons move through it, and as it turns, it produces ATP. It's called proton motive force. And there's what it looks like. I think it's one of the coolest things that I actually didn't get to do any research or study about a lot in college. I think it's one of the coolest things I've ever seen. That you can have these two stages produce things that are going to then fuel this stage, and it can make the abundance of ATP it does without using any energy from energy investment or even substrate level phosphorylation. Literally, it's just the protons slipping in here. And first off, what do you notice about the area that they're in?
They're in
terms of
grooves.
membrane? Is that going to be hydrophobic or hydrophilic?
Hydrophobic.
Hydrophobic. So what are they going to try and do? They're going to try and go away from it. So by this protein first giving them a way of getting in so that they'll eventually get out, it almost tricks them to where they're now attempting to move away from all this hydrophobic stuff, and that results in this protein turning as they're trying to get away from those hydrophobic tails. And it turns and turns and turns until they get to another part of the protein where they can actually slip out and make it back across the membrane. It is. You weren't being facetious. Okay, yeah. I think that's really, really awesome. Okay, here's something completely different real quick, and then I'll let y'all go. We will conclude this. And just so you'll know, this is what the electron transport chain and ATP interface look like together. Okay, this is the whole process used together. All right. So I'll just give you all a cloud and bottle. Where did it go? Try that again. Okay. Tell you what, we'll just play it from where it is.
I don't know what's going on because I don't shield. I don't know what's going on as usual, and I want to see what's happening and learn about it at the same time.
Wow, that's like a student in class. It's perfect.
Yes.
All right, so we're learning about clouds. So I was thinking if you want to make a cloud, you know, the meteorologist talks about high pressure, low pressure, but I wanted to show you things that you can actually relate to. So here's a soda bottle, so it's a plastic soda bottle, and the liquid goes down inside here.
What kind of liquid is that?
Well, we're actually using a mixture of water and some rubbing alcohol because I want it to evaporate really, really quickly, right? So here it is sitting here like this, and now what we're going to do is we're going to pressurize it. So I'm going to take the foot pump and I'm going to put it like this. Got it?
Yep.
And now you're going to simulate a meteorologist, and you are going to put some high pressure. Okay? All right, so good. That's good. Okay, 75 more times. Ready? Here we go. Got it? Got it? Got it? Okay, 50 more times. Here we go. No, it's 5, 4, 3, 2, 1. Perfect. So now, believe it or not, some of that has vaporized, and when I let it go, it's going to go low pressure. Watch this.
Wow. And what caused it to do that?
Because all the water now condensed inside, and so we went from high pressure to low pressure. It's kind of like on the beach, you know, we've had some rain here locally. It's like the fog in the morning. Well, the fog burns off, so watch this. I'm going to put this back inside, and now I just want you to add some pressure. Go ahead and pump it back down again. Look what happens when the higher pressure comes in. Keep going, keep going, keep going. It just vanishes. So you can actually see it kind of vanish. Is that fun?
Yeah.
And then all of a sudden, high pressure comes through, bam, and we got it again.
That's amazing.
Good, right?
All right, guys, have a great day. Remember, quiz on Thursday, only over this chapter and the chapter before. We will finish this chapter on Thursday, and the stuff I covered then will be on that quiz.